Hi and hello, welcome to online biology class because of Ideal&Adharsh Educational Institution and the year. I am Sadish Kumar going to present the topic organization of tissue. So this is the unit number 18 for 9th standard biological science. The continuation of the organization of tissue, we will see about another one important topic that is called the cell division. So we are all known very well. So the small plant or small organism is become into the adult. How it is takes place? Because of only in the cell division. So the cell division is not common into all types of organism, and each and every type of organism they are having different types of cell division. So normally three different types of cell divisions are available. So one is called a mitosis. It is a primitive type of organism. A single organism get divided into two equal organisms in a very fast manner. It is a mitosis. And second one is called mitosis, which is takes place on all the parts of our body cell. And third one is called an meiosis. So the meiosis are two types, which is called meiosis 1 and meiosis 2. So these are the things are seen about today's topic. Before we are enter into this today's topic, we will see some kind of basic animation how the cell division is takes place on each and every type of the organism. So this is the simple and the diagrammatic representation. We will see that this is called a cell. Then cell which consists of cytoplasm and a nucleus. The round blue color substance is called cytoplasm and yellow color substance is called nucleus. So due to the division, the cytoplasm and the nucleus is undergo for dividing and formation of two daughter organism. So there is no death in the parent organism. This is a simple mechanism of cell division. And second one, we will see this is the great animation and presentation regarding the mitosis. Today we are seeing about the two kind of substance. One is called an A mitosis and another one is called a mitosis. So these are the two substances we are going to be seeing. So first phase is called a prophase or interphase. Then followed by the interphase, we are seeing about a prophase and metaphase, anaphase and telophase. Finally, the division of two equal or two daughter cell, that is called cytokinesis. So these are the things are seen about today's topic. Then from the interphase, so it is otherwise called an preparatory phase. The cell before going for the cell division, it was prepare itself. That is called a interphase. And first one we know very well, that is called a prophase. So what is prophase? The nucleus and nuclear membranes are get ready for the division. So prepare itself and undergo for division is called an prophase. Then the chromatins are divided and they are get ready for the division. And second one is called a pro-metaphase. So the formation of spindle fibers, which is move on to pull with the opposite direction. Then they are attached with the chromosome, is called an metaphase. Then followed by the metaphase, a division of anaphase. The anaphase, the spindle fiber pull out to the polar region, dividing of two equal length. And last one is called telophase. So the chromosome spindle fibers are attached and formation of two nuclear cell in the cell. So it is called anaphase, telophase. And last one followed by the nucleus, the cytoplasm also divided. So this is called an cytokinesis. So the division of cytoplasm is called an cytokinesis. So let us enter into the topic for the textual content. And what is cell division? So are you aware that the living organisms start their life from the single cell? Yes. So we are the human beings. We are start from the single cell from our mother uterus. So the egg cell which is penetrated with the sperm cell which is undergo for zygote development. A zygote is a single cell then which is converted into embryo, and embryo is undergo for 2 cell, 4 cell, 8 cell, 16 cell, likewise it is undergo for cell division. So our lives are start with a single cell. Then you may wonder how the single cell them goes for the such large organism. We are seeing that from our mother uterus we are like a very small organism, small ball like structure. Then after 1 week, 2 weeks, second week, third week, then up to 100 days our bodies undergo for various kinds of cell division. The organs are developed. After that 9th month we are become into this young baby, then there comes out from this mother's uterus. This is the development of the organism that starts the life from a single cell. And two more question, what is cell division? All cells reproduced by division, and division of cell into a daughter cell is called a cell division. So reproduction by division, which means the organism get divided into two organisms, and again the division for the cell, so that is called a daughter cell, the continuous division. So there is no death in the parent cell, that is called cell division. And the types of cell division, normally three types of cell divisions are available. One is called a mitosis. This is the direct cell division. Example is amoeba, bacteria. So the single cell is get divided into two cells. The daughter organism is formed without any risk, that is called direct cell division. And second one is called a mitosis, that is called a indirect cell division. So the cell which undergo for various stages: prophase, metaphase, anaphase, telophase, and cytokinesis. After the different phases, which is divided into two cells. And last one is called meiosis. So this is the reduction division. Why we are call upon reduction division, we will see all these things in the next class. Reduction means the chromosomal number is reduced into half, that is reducing of the chromosome number. So it is called a reduction division. And what is amitosis? It is the simplest mode of cell division occur in the unicellular animals, aging the cells to the fetal membranes. So it is a unicellular organism, amoeba, euglena, paramecium, bacteria, yeast, like a unicellular organism. They are done by means of a simple mechanism. So it is called amitosis. And during amitosis, nucleus is elongates first, and construction appear which depends upon the divide of nucleus is 2. Then followed by the cytoplasm divides, result in the formation of two daughter cell. So this is the diagrammatic representation. A cell which consists of a cytoplasm, nucleus, and cell membrane. Then followed by first division takes place on the nucleus. The nucleus is get enlarged. That is called construction. Followed by the enlarge of the nucleus, the cytoplasm also get construction. Then finally what it will be happen, due to the process of cytokinesis, a single cell get divided into two daughter cell. So this is the simplest mechanism which is takes place on all types of unicellular organism. And we can move on to the another type of cell division, that is called mitosis. It was first discovered by Fleming. So the Waldeyer Fleming, he is a person who was discovered the cell division. So Alexander Fleming, another one person is that. This is a different Fleming in the year of 1879. One more question. This is the cell division. One parent cell divided into two identical daughter cells. That means similar to their own offspring, each with the nucleus having the same amount of DNA, which means 46 chromosome. Then daughter cell also consists of 46 chromosome. Then same number of chromosome in the gene present in the parent. So it is called an equitotional division. So equal number of chromosome. We are all having a 46 number of chromosome or 23 pair of chromosome. Our parent also consists of 46 number of chromosome. So it is a equitotional division. And mitosis consists of two events. One is called karyokinesis, another one is called cytokinesis. Karyokinesis, that means a chromosome which undergo for division. A chromosome which undergo for division is called karyokinesis. Cytokinesis, which means a cytoplasm undergo for division depending upon the nucleus. Is called an cytokinesis. Let us enter. So we will see that a diagrammatic representation, karyokinesis and cytokinesis are given. And the upper picture you will see that the Fleming, who was discovered the mitosis process. And then before we will see that the cell prepare itself, that is called interphase. So it is a resting phase of the nucleus, the interval between the two successive cell division. Before that, we will involve for any kind of competition or prepare ourselves, and we will take some rest like that. The cell also getting some kind of rest position for producing their new cells, that is called interphase. Then the cell prepare itself for the next cell division. Before that, the cell was prepare itself for division, is called interphase. So this is the diagrammatic representation of interphase. Then centrioles, chromatin, nucleus, nuclear envelope, plasma membrane, everything is present on this cell stage. And karyokinesis, the division of the nucleus into the daughter nuclei is called karyokinesis. A nucleus get divided is called karyokinesis, which is 4 phases: prophase, metaphase, anaphase, telophase. PMAT, PMAT, prophase, metaphase, anaphase, and telophase. Let us see that what are the stages of cell division. This is the overall representation of the cell division. Interphase, interphase to prophase, prophase to late prophase, late prophase to metaphase, metaphase to anaphase, anaphase to telophase, and finally the cytokinesis process, which was end of the process. We are get down with the 2 daughter cells. Now we will see that prophase. Pro means first. It's a first division, so it is called a prophase. During this stage, chromosome become shorter and thick, clearly visible inside the nucleus. Then centrosome splits into the centrioles and occupy the opposite pole of each. Centriole is surrounded and is asterase. Spindle fibers appears between the 2 centrioles. So nuclear membrane and nucleolus are disappear. We will see that the nuclear membrane and nucleolus are disappear. Nuclear envelope are formed. Centriole attached with that centromere and separating of 2 centrosome, and mitotic spindles are formed with inside of the nucleus. So this is called prophase. And next one we will see that metaphase. Then after the prophase, so it is called metaphase. So duplicate number of chromosome. So 1 chromosome is get divided. The 2 chromosome become 4 chromosome. The arranging of equal plane from the metaphase plate. Each chromosome get attached into this spindle fiber for the centromere. Then centromere of each chromosome divides into 2 each beginning associate with chromatids. We will see that the polar microtube is called centromere. Then the centromere attached with spindle fibers, a thread-like structure is called spindle fibers. They are pulled with opposite direction. So the centromere is pulled with opposite direction. That is antipole region. The chromosomes are get divided, which is try to move on with opposite direction. It's called metaphase. And we will see about the third one is called anaphase. The ana means up and back, up and down or up and back. So one chromosome is move on with the polar region and another chromosome is move on with another polar region. That is a top region, bottom region. The centromere attach the 2 chromatids divided into 2 daughter chromatids. Each chromosome separate and migrate towards the 2 opposite poles. I already told you upper pole and down pole, up pole and down pole. The chromosome and centromere are attached with the spindle fibers, which pull by means of pressure and which is move on with the 2 opposite poles, which is called an anaphase. Then anaphase we will see here the chromatin and centromere, which is move on with the 2 opposite direction, increasing the separation of spindle poles, which is not given in your textbook. Please go through all these things in this diagram. And last one, that is called telophase. Tel, that means end. So come to the end. Then each chromatid or daughter chromosome lengthens and become a thinner and turns into the network of chromatin threads. Spindle fibers are broken up and disappear. Then nuclear membrane and nucleus reappear and daughter cells are formed. So the nuclear membrane, nucleus is reappear, spindle fibers are broken and the move on with the opposite fold. So now the 2 new nucleus are get ready. That is called a daughter nucleus are get ready, and which is undergo for cytokinesis. So the last process, the cytokinesis, the division of the cytoplasm into the 2 daughter cells by the construction of the cell membrane is called a cytokinesis. 2 mark question. Division of the cell membrane depending upon the nucleus is called a cytokinesis. Why cytokinesis process takes place? Without cytokinesis, the cell which is not surveying. So the cytokinesis which was surrounded with the nucleus. So the cytokinesis process takes place. And this is a cytokinesis process. Then 2 nucleus and centrioles are present, and due to the construction ring which takes place on the cytoplasm, formation of 2 daughter cells. And this is a overall representation of the mitosis. First one is called interphase, prophase, metaphase. Then we will see that anaphase and telophase begin with the cytokinesis are given in this diagrammatic representation. And we will see that the significance of mitosis 5-mark question. This is equatorial division, we know very well. Then diploid daughter cells, which takes place on the diploid daughter cell. So 2n, that is they are having a two different types of nucleus, so it is called 2n with the equal distribution of genetic material. So all the human being, they are consist of DNA as a genetic material, except the retrovirus. All other organisms, they are consist of DNA as a genetic material. Then exception of this retrovirus, they are having a RNA as a genetic material. Then second significance, the multicellular organisms growth, organ development and increase the body size accomplished through the process of mitosis. I already told you from the beginning. Then our growth, development of organ, increasing the size, everything takes place by means of mitosis. Then mitosis help to repair damage and wounded tissue by the renewal of loss of tissue. Then we are fall down, we are getting a wound or any kind of loss of our body cell, we are make the suturing. So all the cells are renewed by means of a mitotic cell division. So then up to that we will successfully seen about the cell division introduction and see about amitosis and mitosis. Next class we are seeing about the interesting topic of meiosis and the types of meiosis. Then they are having two types and meiosis 1 and meiosis 2. These are the things are seen about the next class. Up to that, thank you, bye bye from Sadish Kumar from Biological Science Department.